Good morning, everyone, and welcome. I am delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, I will be talking about the calibration of high-performance liquid chromatography, or HPLC systems. This is a crucial topic for ensuring accurate and reliable analytical results. Over the course of this presentation, we will explore the key steps involved in calibrating HPLC systems, discuss best practices, and highlight common challenges. I look forward to sharing valuable insights with you all. Let's get started. Today, I'll walk you through the main objectives and scope of calibration in HPLC systems. Our primary goal is to describe the key calibration steps necessary to verify the performance of your HPLC, including accuracy, precision, linearity, flow rate, and detector response. These are essential parameters to ensure your system is producing reliable and reproducible results. The scope of this calibration process applies to all HPLC systems used in both quantitative and qualitative analysis, especially in regulated laboratory environments. Whether you're analyzing pharmaceuticals, environmental samples, or other complex mixtures, these calibration steps are fundamental to maintain compliance and confidence in your data. Now, let's look at the components of the HPLC system that require calibration. First, the pump, which controls both flow and gradient. Ensuring its accuracy impacts everything from separation to quantitation. Next is the auto sampler, specifically the inject volume, which must be precise to guarantee consistent sample introduction. The column oven's temperature needs calibration as well, since temperature fluctuations can affect separation efficiency and retention times. Finally, the detector itself, which requires calibration for linearity and wavelength accuracy. Proper calibration here ensures that your detector's response is proportional to analyte concentration and that the wavelength used for detection is accurate. When all these components are properly calibrated, you can confidently verify that your HPLC system is performing optimally, which is the core focus of our calibration process. Let's take a closer look at the regulatory framework that guides our analytical practices. First, we have USB 1058, which covers the phases of analytical instrument qualification, namely the design qualification, installation qualification, operational qualification, and performance qualification. These steps are essential for ensuring our instruments are properly installed, functioning correctly, and consistently producing reliable data. Next, USB 621 focuses on chromatography, emphasizing the calibration of detector linearity and the use of both external and internal standards. This helps us maintain accuracy and reproducibility in our separation methods. Then we have ICH-Q2, R1, which provides guidelines on key validation parameters like accuracy, precision, and linearity, especially for quantitative methods. These parameters are fundamental to demonstrating that our analytical methods are fit for purpose. Finally, 21 CFR 211, 160, B. 4 specifies the required intervals for instrument calibration, ensuring ongoing reliability over time. All these references collectively form a robust regulatory backbone that underpins our quality assurance efforts and helps us deliver consistent, compliant results in our work. Let's take a closer look at acceptance criteria general. These criteria are really the foundation for defining what successful completion looks like for any project or task. They serve as a clear set of conditions that must be met, so everyone on the team knows exactly what is expected. By establishing these criteria early, we can ensure alignment and reduce misunderstandings down the line. It's important to remember that acceptance criteria should be specific, measurable, and testable. This way, we can objectively evaluate whether the work has met the set standards. Connecting this to our overall theme, having well-defined acceptance criteria helps us deliver quality results efficiently and confidently. It keeps the project moving forward smoothly and ensures stakeholder expectations are properly managed. So, in essence, Good acceptance criteria are key to successful project delivery and continuous improvement. Let's take a closer look at calibration step 1, flow rate accuracy. This step is crucial because ensuring that our flow rate is accurate directly impacts the reliability of the entire measurement process. When we talk about flow rate accuracy, we're essentially verifying that the fluid or gas passing through our system matches the expected or specified rates. If the flow rate is off, it can lead to errors in our data and compromise the overall quality of the calibration. So, during this step, we carefully measure and compare the actual flow rate against the standard or reference values. This helps us identify any discrepancies early on. The key concept here is precision, making sure every measurement is as close to the true value as possible. Connecting this to the broader theme of calibration, maintaining flow rate accuracy is fundamental because it sets the foundation for all subsequent calibration steps. Without accurate flow rates, other parameters might seem correct, but in reality, they could be skewed. Overall, this step ensures that our system is operating as it should, providing a reliable baseline for the rest of the calibration process. Now, let's take a closer look at our linearity data and plot. This slide focuses on the calibration step, specifically step 2, which involves injection precision and linearity. First, we'll discuss precision or repeatability. To evaluate this, we inject a standard solution, say 100 micrograms per milliliter of caffeine, 6 times in a row. We record the area for each injection and then calculate the relative standard deviation, or RSD, to see how consistent our injections are. This helps ensure our method is reliable and precise. Next, we move on to linearity preparation. Here, we prepare standards at different concentrations, like 10, 20, 40, 60, and 80 micrograms per milliliter, and inject each in duplicate. By doing this, we can assess how well the response, meaning the area, is proportional to the concentration across our range. This step is crucial because it confirms that our method can accurately measure samples within that concentration range. Overall, these steps, checking precision and linearity, are key to validating our analytical method, ensuring our results are both accurate and reproducible throughout the calibration process. Now, let's take a closer look at how we evaluate detector linearity. For each level of caffeine standard, we inject it twice to ensure consistency, and we record the mean peak area for each injection. This duplicate injection helps us verify that our measurements are reliable and reproducible. In this example, we're using a UV detector set at 273 nanometers, which is optimal for caffeine detection. We inject 10 microliters of the sample, and our mobile phase is a mixture of water and methanol in a 60-40 ratio. The flow rate is maintained at 1 milliliter per minute to ensure stable and consistent flow through the system. The main objective here is to confirm that the detector's response, the peak area, is directly proportional to the caffeine concentration within our working range. This proportionality is crucial because it underpins the accuracy of our quantification. When we talk about detector linearity, we're essentially checking that as we increase the caffeine concentration, the detector's response increases proportionally. If this relationship holds true across the range of concentrations we're working with, it validates the reliability of our analytical method. This step is fundamental because it ensures that our measurements are accurate and that any quantification we do is based on a solid, linear response from the detector. It ties directly into the overall accuracy and precision of our analysis. Let's continue our discussion on detector linearity. As we saw earlier, maintaining a linear response is crucial for accurate measurements. On this slide, we're focusing on some additional aspects that influence linearity. First, it's important to recognize that even small deviations from linearity can significantly impact the quality of our data, especially when dealing with subtle signals. We need to thoroughly test the detector's response over the entire range of expected signals to ensure it remains linear. Furthermore, calibration plays a vital role here. Regular calibration checks help us identify any nonlinear behaviors that might develop over time, allowing us to correct them before they affect our results. Also, note that temperature stability and electronic noise are factors that can influence linearity, so controlling environmental conditions is essential. Connecting this to our overall theme, understanding and maintaining detector linearity ensures the integrity of our measurements throughout the experiment. It's about making sure that what we see truly reflects the phenomena we're studying, without distortion or bias introduced by the equipment itself. Now, let's take a closer look at the acceptance criteria based on the USB and ICH guidelines. First, we have the R-squared value, which needs to be greater than or equal to 0.999 to pass. This indicates an excellent linearity in our calibration curve, essential for accurate quantitation. Next, we perform a visual inspection of the straight-line plot. If the plot appears linear and consistent throughout the range, that's also a pass. Moving on to detector linearity, which is crucial for ensuring our instrument's response is proportional to analyte concentration. Remember, these criteria are interconnected. They help us confirm that our method is reliable and reproducible. By meeting these standards, we can confidently proceed with our analysis, knowing our calibration curve and detector response are within acceptable limits. This consistency is vital for the overall quality and integrity of our results. Today, I want to focus on our main objective for this part of the process, which is confirming that the actual wavelength emitted by the detector matches the set wavelength. Essentially, we're verifying that the instrument is providing accurate readings. To illustrate this, let's look at an example involving wavelength accuracy using a holmium oxide filter. Holmium oxide is particularly useful because it has certified absorbance peaks that are traceable to recognized standards. This traceability is crucial because it ensures that the peaks we observe are reliable and can be used to validate the wavelength calibration of our instrument. The goal here is to ensure that the wavelength we set on the device corresponds precisely to what the detector actually measures. This helps us maintain accuracy and confidence in our results throughout the entire process. Let's continue our discussion on wavelength accuracy. As we've seen, maintaining precise wavelength measurements is crucial for many applications, but it's an ongoing challenge. In this slide, we'll explore some of the factors that influence wavelength accuracy and how we can address them. First, environmental conditions like temperature, pressure, and humidity can cause the physical properties of our measurement systems to change, leading to inaccuracies. That's why controlling or compensating for these variables is so important. Second, calibration plays a key role. Regular calibration against known standards ensures that our instruments stay accurate over time. Without this, small drifts can accumulate and significantly impact our results. Finally, technological advancements are helping us push the boundaries of wavelength precision. Improved laser sources, better detectors, and more sophisticated calibration techniques all contribute to more reliable measurements. Connecting this to the overall theme, achieving high wavelength accuracy is fundamental for ensuring the reliability of our measurements, whether we're working in research, manufacturing, or quality control. It's about understanding and mitigating factors that can introduce errors, so we can trust our data and make informed decisions. Now let's talk about auto-sampler calibration, specifically focusing on injection volume accuracy and precision. To ensure our results are reliable, we start by using a reference standard solution. The key here is to inject the same volume multiple times, say 10 microliters, 6 times in a row to check consistency. Next, we calculate the mean area of those injections and determine the relative standard deviation, or RSD. For example, if the mean area is 50,000 and the standard deviation is 400, then the RSD would be 0.8%. This tells us how consistent our injections are. Importantly, a pass criterion for this check is an RSD of less than or equal to 2%. This ensures our injection process is both accurate and precise, which is crucial for the overall reliability of our analytical method. Remember, maintaining this level of precision helps us trust that our results are reproducible and accurate across runs. Let's talk about the system suitability check. According to USP guidelines, this step is crucial because it verifies that our analytical method is ready for routine use. It essentially confirms that our system is performing adequately before we move on to analyzing samples. Key parameters we look at include resolution, which tells us how well our peaks are separated. This is vital for accurate identification. Then there's precision, often expressed as RSD or relative standard deviation, which should be less than or equal to 2%. This ensures our method yields consistent results. We also evaluate the tailing factor, which should be less than 2. This indicates that our peaks are symmetrical and not fronting or tailing, which can affect quantitation. Additionally, we consider the theoretical plates, reflecting the efficiency of our chromatographic column. More plates mean better separation. It's important to note that these criteria are not just theoretical. They need to be met during calibration runs. Meeting these standards confirms our system's readiness and paves the way for reliable, reproducible results throughout our analysis. Now, let's talk about out-of-tolerance, or OOT, handling. When results fall outside the accepted range, it's crucial to take specific steps to address the issue properly. First, we need to tag the instrument to identify that there's a potential problem. Then, it's important to investigate the cause of the out-of-tolerance result. Understanding why it happened helps us prevent future occurrences. Next, we assess the impact this result may have on the affected data, ensuring we understand any potential implications. After that, we recalibrate the instrument once the cause has been corrected, which helps restore accuracy. Finally, all of these actions must be documented as part of the corrective and preventive actions, or CAPA, process. Remember, OOT events are major inspection issues because they can directly impact data integrity and product quality. Handling these properly is essential to maintaining compliance and ensuring reliable results throughout our processes. Let's talk about best practices for calibration. First, it's crucial to always use certified reference standards. These standards ensure the accuracy and traceability of your measurements. Maintaining standard operating procedures, or SOPs, is equally important because they provide consistent guidance for your calibration processes. And don't forget to train your analysts thoroughly. Well-trained staff are key to reliable calibration results. Now, when it comes to calibration frequency, there are some suggested intervals to keep in mind. For HPLC systems, a full calibration should typically be performed every 3 to 6 months. Of course, you should also recalibrate after any major repairs to ensure everything is functioning correctly. Additionally, before running critical study samples, it's a good practice to do a calibration check to confirm your system is still accurate. By following these intervals and best practices, you help ensure the reliability of your analytical results. This consistency is fundamental to maintaining quality and compliance throughout your laboratory processes. Let's talk about documentation and records, which are essential parts of our calibration process. When preparing for calibration, it's important to have a few key documents in place. First, we need an approved calibration protocol that clearly outlines the procedures and acceptance criteria. This ensures everyone is on the same page and the process is standardized. Next, we should keep raw data worksheets. These are the detailed records of the calibration measurements taken during the process. They provide the evidence needed to verify that calibration was performed correctly. We also need to have calibration certificates on hand. These certificates confirm that instruments meet the required standards and are properly calibrated. Additionally, it's crucial to document instrument ID, the date of calibration, the analyst who performed the work, and the reviewer's signatures. This traceability is vital for accountability and audit purposes. And remember, all of these records should follow the ALCOA plus principles, which emphasize data being attributable, legible, contemporaneous, original, and accurate, plus the additional principles of complete, consistent, enduring, and available. By maintaining thorough and compliant documentation, we uphold data integrity and ensure the reliability of our calibration activities, which ultimately supports the quality of our work and compliance with regulatory standards. For further support, please contact us. I'm happy to help with any questions or additional information you might need. And with that, I want to sincerely thank you all for your attention and engagement today. If there's anything more you'd like to discuss, don't hesitate to reach out. Remember, our goal is to ensure you feel confident and equipped with the knowledge shared. So again, thank you very much.